So let's start with this. Now this is just a reflection that means of what we are going to learn today. We are going to learn that what is plasmids. There are various type of plasmids. I mean to say the cloning vectors. So these are the kind of vectors, okay, cloning vectors, which is plasmid 1. So we will learn the different type of plasmid, that how to extract the target gene, that means of which type of suppose if you want to extract the desired gene, so how you will extract the desired gene from a particular organisms, that we're going to learn from any cell. Like you, if you have multicellular or unicellular animals or organisms, so you can extract the target gene and you can insert it into the plasmid vectors. And after insertions, how you will join this, like here you can see target gene and this is the vector. So how you will join this foreign gene into this plasmids. So there is a process that is called ligations, and then this will be ready. So what are the process, that how it start, we will learn in details about this technology and tools. Now here is just to recall the basic concept of rDNA, like how to do a multiple cloning of particular desired gene. Like here you can see this is a particular cell of your interest. Like if you want to you are interested in any bacterial cell or any animal cell, so you can just extract the gene of your interest from that cell. That is your first part. Then you have to select the most appropriate vectors, and that vector can be a bacterium. So you know, inside the bacteria is a plasmid, so plasmid can be act as a vector. So you can just cut the vector at the desired point, and for cutting that vector you can take a help of enzymes. Can you tell me that what is the name of that enzymes which we can take and cut the DNA, or any cut the DNA plasmid vector at the desired site? Yes, just tell me the name of that enzymes which act as a molecule, which also called it as a molecular seizures. Yes, restrictions enzymes. But you know that in restriction enzyme there are total 3 type of restrictions enzymes, right? Exonuclease, endonuclease. Yes or no? Yes, endonuclease, right? So yes, it's endonuclease because you're cutting the specific points, right? So here you can see the vectors are, I mean to say, plasmids cut at the specific point, and then you can join this gene of interest into this plasmids, and this joining is take place by mean of ligation enzymes. Like there is a ligation enzymes through which you can join, and again you have to insert this recombinant DNA plasmid into the bacterium cell, and then this bacteria start multiplying. So the number of clone cell will be increased in number. So this is what the cloning process, which we have already studied, but in very basic, not in detail. And so we'll talk about more details about it, because in the previous session we studied about the various enzymes, a detail we studied very details of enzyme, that what kind of enzymes will help for restrictions, and different type of enzyme we studied. Today we will learn the next step, that what are the type of vectors available in the nature, and that vector can act as a multiplication, right? So we will talk more about it. So let's connect with this real life. So you know how vector actually work. So vector, you know, similar kind of vehicles, any kind of vehicles. Like we can say, suppose any manufacturing unit, a pharmaceutical company, okay, who preparing medicine. Hello, yes, anyone want to speak? No, okay. So a kind of pharmaceutical company, or we can say manufacturing company, who want to deliver their product into their warehouses. So they need a certain vehicle, or we can say that transporting material. So that vehicle is generally used to deliver the desired project at the desired locations. Similarly, as the vehicle work here, vector is also work the similar concept. So it is, we can connect this with the real life examples, like how this cloning vector work. Is this clear to everyone? Yes. Okay. Now here, our actual cloning vector start. Now, uh, first of all, we need to understand that what is this cloning process and all. So very first of all, why we need a vector? What is vector? So vectors are the DNA molecule. Simply, I can say that vector is what? Vectors are the DNA molecule that can carry a foreign DNA segment. What is mean of foreign DNA segment? That is your desired DNA segment. Like here you can see this is the bacterium, and this blue one indicate your desired gene. Okay, and this yellow one is the vector. Okay, so vector is what? Vector are the DNA molecules that can carry the foreign DNA segment of your interest and replicate inside the host cell. So what is the host cell? Here you can see the bacterium is a host cell. So you inserted this into the host cell, and this host cell bacterium have capacity to divide or multiply by binary fission. So you can see the multiplication number is going to increase 2, 4 and followed by 8, right? So this number is going to multiply and huge copies can be created with this bacterium. So that means of these plasmids having the huge copy. Initially it is just one copy, but you can see there are total n copies can be created from this. So actually vector is work as a vector is what? It is a DNA molecule that carry a foreign DNA segment and replicate inside the host cell. Now, this vector may be plasmid, bacteriophage, cosmid, artificial, yeast artificial chromosomes, even the bacterial artificial chromosomes, and viruses. That means a vector can be related to plasmids. How you can say this is a vector? The vector can be a plasmid. It can be a bacteriophage. You know bacteriophage. What is bacteriophage? Any idea about the bacteriophage? Bacteriophage. Why it is called bacteriophage? Because bacteriophage is a kind of virus that infect the bacteria. Very good. And it is a kind of virus who basically infect the bacteria, and that's what is called bacteriophage. Clear? Yes, Jason, is this clear? Okay, so these are the name of certain vectors. Let me write here certain vectors name is, the first one is plasmid. Plasmids act as a vector. Then there is a bacteriophage. Bacteriophage. Another one is artificial cosmids is also there. Cosmids. Then yeast artificial chromosome, bacterial artificial chromosome, that is yeast. So it is short form I have written, that is YAC and BAC. So it's a chromosome. So SS you can write it here. So these are the vectors. Even some virus can also work as a vector. Some viruses is also work as a vector. So these are the specific vectors who can work for cloning, work as a cloning vectors. Clear? So you have to remember the plasmids, bacteriophage, cosmids, YAC, BAC, and viruses. YAC, BAC is what? It is a yeast artificial chromosomes, and BAC is a bacterial artificial chromosomes. Clear? Okay. So cloning vectors are structure that help in the making multiple copy of desired DNA fragment. So it is, you have to remember what is cloning vector. So name is cloning vector, means you can multiply your DNA fragment, your desired DNA fragment in n number. So multiply the copy of desired DNA fragment n in number, that is what called the cloning vector. Now we have very much clear about what is vector and what is cloning vector. Clear? Now let's move ahead. So this vector can replicate within the host bacterial cell. if it is replicated in the host bacterial cell, like any bacterium, some of them are present naturally in the bacterial cell, while in others they can be introduced artificially. And introduced artificially, that is YAC and BAC, which is introduced artificially, and that's why the name itself, that bacterial yeast artificial chromosomes. Why said artificial? Because some are introduced by the by the humans, and that's why it is called the artificial. So it can be naturally in the bacterial cell, or while others can be introduced artificially. So introduced artificially is YAC and BAC is the most common example you can consider. Now, let's understand what are the characteristics features of these cloning vectors. Like, as you know that what is mean of cloning vectors. So, if you select any cloning vector, what are the basic features should be there, right? So, in any cloning vector, the four important features should be there. The first feature is called the origin of replication, or it is also in short it is referred as a ORI. ORI is what? Origin of replication. This is in short it is referred as ORI, origin of replication. Now this origin of replication is a specific sequence of DNA bases which is responsible for initiating the replication. Means this point you can see, find in this is a complete vector, cloning vector. Can you able to find where is the origin of replication? Here, where is the origin of replication? You got it? Yes. So you can see here, this is the origin of replications. Means in this entire vector, this particular location is referred as a origin of replications, where replication is going to start at this point only. Okay. So in any cloning vector, there should be a ORI. ORI is what? Origin of replication. Clear? Now, what is the role of this? So basically, when I say this ORI is a specific sequence. This is also very important. It is a specific sequence. Sorry. It is what? It is a specific sequence of DNA. ORI is what? It is a specific sequence of DNA which is responsible for initiating the replications, and this is responsible for initiating what? Replications. And you know the meaning of replication, because now you should know very well what is replications. Replication means to multiply in number. If you have one copy, you can create n copy through this process, which is called replication. That means of this origin is very specific, and it is specific of the sequence of DNA, and this origin is responsible for initiating the replications. Now let's move ahead. Now in prokaryotic DNA, this is what I talk about the eukaryote. Now, but in prokaryotic DNA, it has a single origin of replications, while eukaryotic DNA may have more than one of the replication. This is also very interesting. Okay, so how you will identify prokaryotic and eukaryotic? So in prokaryotic, there is only one single replication is there. Single replication is there. Origin of replication is there. So, and in eukaryote, there is a multiple replication site is there. Multiple origin of replication is there. So can you, based on this statement, can you tell me that this image represent belongs to prokaryote or eukaryote? It is prokaryotic, right? Vector, why? Because it has only one replication point. If there are several replication origin like here and here also, then we can call it as a eukaryotic. Clear? This is called the eukaryotic DNA. Clear? Okay. Now let's move with the another one. So yes, anyone have a doubt on this origin of replication? I want everyone to confirm in the chat box. Kindly use the chat box and confirm. Anyone have a doubt? Okay. Okay. So, and Jason and Bhuana responded. I want response for others also like Sarjana, then Gopinath and Ragul. Sarjana, Gopinath, Ragul, please respond it.
Sir, no doubt, sir. Sir, actually, I already responded, sir.
You responded? Okay,
Yes,
let me check. Oh, yes, got it. Okay, so remaining, please respond it. Okay, Ragul responded. One more is there. The name is Gopinath. Please respond it fast, so I can go ahead. Gopinath. Okay. Now the next is selectable marker. Okay. First is origin of replication. Second is selectable marker. Now, what is this selectable marker? So first of all, you have to locate in this vector where is selectable marker. So here you can see this particular point, quite difficult to draw. Okay, so here you can see selectable marker in the same vector. Okay, so in the same vector, another characteristic feature is they have a selectable marker. Now what is the selectable marker? So some gene called selectable markers in that plasmids, right? Some genes are called selectable marker, and it is helping in selecting those host cells which contain the vectors. Like simply I can say transformant, right? And also it eliminates the non-transformant, means those who can able to transform, they are called transformant. So, and those who cannot what we can say transformed, then it is eliminated from the non-transformants. Clear? So it is transformation is what? It is a kind of process through which a piece of DNA is introduced into the host bacterium. That is called transformation. So please keep this important point. Selectable marker is transformant. Formants. Now what is mean? That means of transformation. Transformation is what? It is a process through which a piece of DNA is introduced into the host bacterium. Okay, that is important. So it is the piece of DNA. DNA introduced into host cell. Clear? Host cell of bacterium. Okay. Bhuvanasri said it helped to identify the transformers from non-transformant. Yes, that you can see. That means basically because transformers is required, so it also helping in removing that non-transformant, like those who is not required, they can easily eliminate it by this selectable mark, right? So it is used. Now generally the gene encoding resistant of antibiotics, like such as we can say tetracycline, ampicillin, kinamycin, chloramphenicol, These are useful selectable marker for especially for E. coli bacterium. Okay, let me write here the name of some selectable marker, and that you have to also remember the important one. The first one is tetracycline. Okay, you have to remember this one. The first is tetracycline. Tetracycline. Another one you can see ampicillin. Ampicillin. Third one, that is called the kinamycin. Okay, you can say kanamycin or kinamycin. The fourth one, that is called chloramphenicol. Okay, so these are called the selectable markers. Yes, so you have to remember the name. Now these selectable markers are useful selectable markers, especially found in E. coli bacterium. And the common E. coli cells are not resistant against any of these antibiotics. So the most common, so plasmid PBR322 has two resistant gene. Like in plasmid PBR, they have two important resistant gene, that is called ampicillin and tetracycline, and which are considered useful for selectable marker. So that means of in E. coli there is PBR322. So in PBR322 there is two important selectable marker. One is ampicillin and another one is a tetracycline. Okay, that is work as a selectable marker. Now go for the next one. The third characteristics features for this cloning vehicle or cloning vector is the restriction site or recognition site. Okay, this is the third one. Restriction site or recognition site. Where is the restriction site or recognition site? You can see here this portion is called restriction site or recognition site. Now what is the role of this recognition site? This recognition site is also called the cloning site. This is also called the cloning site. Okay, now this cloning site, especially in case of, I can say, plasmids 322. So in this plasmid 3 PBR322 has a variety of unique recognition site for restrictions endonuclease. So two unique site is called the PST first, and another one is called the PVU first. These are the two unique site. It is a short, what we can say, short name of that restriction site which is referred, right? So you can call it as a name itself as a PST first, and PVU are the located within the ampicillin gene, and that can be written as small AMPR, like that. Okay, so these are the different codes written based on their site. So these are the cloning recognition sites. Some other unique restriction sites are like there are eco R1 in E. coli. So eco R first. Then there is HINT3. Okay. Some others are like we can say PUV2, PVU2. These are all are the cloning site. Okay, or we can say restriction site, or we can say recognition restriction site especially. Clear? So this is all code for proteins which involve in the replication of the plasmids. Is this clear? Okay, let me tell you the very interesting fact about this cloning site recognition, especially PBR plasmid PBR 322. The very interesting fact is that the presence of restriction site within the markers, like I can say the marker can be tetracycline or ampicillin. It can be tetracycline. So in short, I can write TET. So that is stand for tetracycline R. And the another marker is AMP, small AMPR, that is called ampicillin R. These are the two restriction sites that permit an easy selection of cell transformed with the recombinant PBR 322. That is a plasmids. Now here insertion of DNA fragment into the plasmid using enzyme PST1 or we can say PVU1 place our DNA insert within the gene of ampicillin. Means it is based on the certain selectable markers, right? That selectable markers generally work in bacterial cells which contain such a recombinant will be available to grow the presence of ampicillin, and but will grow on the tetracycline also. Similarly, when the restrictions enzymes, especially we can say BAM H1 or we can say SAL1 is used, the DNA insert is placed within the gene of tetracycline, making it non-functional. That means of each and every selectable markers are very specific in certain cases. And here bacterial cells that basically possessing such a recombinant will therefore grow on ampicillin but not on the tetracycline. That is also observed in some cases. So due to this inactivation of antibiotics, because all these are tetracycline, ampicillin, these all are the antibiotics. So due to some are active, some are inactive. So due to this inactive of antibiotics, selection of recombinant become burdensome, and that process because it requires simultaneous planting on two plates having different antibiotics. And because of these conditions, alternative selectable marker is developed to a different recombinants and non-recombinant on the basis of their ability to produce the color in the presence of chromogenic substances. Right? So based on that, now it is very simple to detect in recombinant DNA is inserted in the coding sequence of enzymes, and that coding sequence of enzymes is called beta galactosidase. Beta galactosidase. Galactosidase. Now, what is this beta galactosidase? It is the recombinant DNA which is inserted in the coding sequence of enzymes called beta galactosidase. So beta galactosidase is a coding sequence, right? And this causes inactivation of enzymes which is called the insertional inactivation. If the plasmid in the bacterium does not have an insert. So the presence of chromogenic substrate, give blue colored colonies, and the presence of insert result into the insertional inactivation of beta galactoside, and therefore these colonies do not produce any color, and this is called marked as a recombinant colonies. So it is the beta galactoside who is code for particular enzymes. That is called the coding sequence of an enzymes. So this is how the selectable, the recognition site basically work for the cloning site, or we can say the recombination site. Yes, so that is what the another one Vector for cloning gene in the plants and animals. Now, vectors of cloning gene in plants and animals. So you know that plant means it related to soil. So a soil inhibiting plant bacteria, like most of the soil having this bacteria which is called Agrobacterium tumefaciens. Agrobacterium tumefaciens. This is generally found inside the plant. Let me write here. This is called Agrobacterium agrobacterium. It's a tumefaciens. Yes, it's a tumefaciens is the name of vector. I mean, say it is a kind of bacterium which is present inside the soil. Now, a pathogen of several dicot plant is able to transfer the piece of DNA known as tDNA. tDNA. Known as tDNA. Please remember, this is known as tDNA. Now, the tDNA cause tumors, and that's why it is called tDNA. T stand for tumors, tumor causing DNA. So, T stand for tumors. So the tumors are called the crown gall. In plant, this tumor, okay, these tumors cause crown gall disease. Crown galls. Okay. Now the tumor formation is induced by a certain plasmid, and the name of that plasmid is induced by TI plasmid. Okay, it is induced by TI plasmid. Now here TI plasmid, the tumor formation is induced by the TI plasmid, and as the gene transfer occur without human effort, the bacteria is called the natural genetic engineer of the plants. Okay, because it is automatically happening in the nature, so that's why there is no human involvement in this transferring of tDNA in the host plant. That's why it is called the natural genetic engineering of the plants. Bacteria is called it's the engineer of the plants. So that bacteria called natural genetic, it is genetic engineer of plants. Clear? So similarly, so this is about the one case. Similarly, there is a retroviruses which cause the leukosis or sarcoma type of cancer in animal, including human also, are able to change their normal cells into the cancerous cell. So a tumor including TI plasmid of Agrobacterium tumefaciens have been modified into the cloning vectors, which is not a pathogenic to the plant. However, it is still able to use the procedure to deliver the gene of our interest into the various plants, right? So retrovirus are used to carry the desirable gene into animals, and that's why this gene or the DNA fragment is joined to suitable vector, which is transformed into the bacterial plant or animal host where it undergo multiplication. So in animal case, it is used as a retrovirus, not rotavirus. So please keep in mind, in plant it is called the TI plasmid, and in animal it is a retrovirus. Retroviruses. Clear? So this is the four important characteristics of the cloning vectors. Now, as I told you that this cloning vector may be plasmid, bacteriophage. So there are various type of, out of which plasmid, bacteriophage, cosmids, then YACS, bacterial artificial chromosomes, BAC, and biases. And there are some subtle vectors also present in the nature. So we will see that each and every vectors, like their capacity and how of gene of interest can able to transform into multiple copies. Okay, that we will also learn. So let's focus on what are the types of cloning vectors. So the very first is a plasmids. Plasmid is what? It is a vector. Correct? So plasmid is a kind of vectors were discovered by the William Hayes and Joshua Lederberg is the name of that discoverer who first time discovered these plasmids. Okay, so plasmid is what? It is a extra chromosomal. Simply, plasmid vector is what? It is an extra chromosomal, self-replicating. This is very important. Extra chromosomal, self-replicating, and generally circular in nature, double-stranded DNA molecule, which is found naturally in any bacterium or also some yeast cell. These plasmid vectors are found. Means it is found in all type of bacteria, and it is found in some fungi like yeast. Clear? That means of this plasmid vector is found everywhere in bacteria and yeast. Now, the plasmids are usually not essential for normal cell growth and division. They often confer, some trait on the host organisms. For example, I can say resistant to certain antibiotics or toxins that can be selective advantage under the certain conditions. So, plasmid molecule may be present as one or two copies in the multiple copies inside the host organisms, and then naturally occurring plasmids have been modified to serve a vector in the laboratory. So the most widely used, versatile, and we can say easily manipulating vectors are PBR322. This is most common plasmid vector. The name is PBR322. Is this clear? Yes. Yes, respond in the chat box, all of you. Any doubt in this? Okay, who is going to tell me the characteristic features of cloning vectors? Tell me. Who is going to tell me the basic characteristic features of the cloning vector? What are important features of any cloning vector? Yes. Four important features, please tell me. At least you can say the important points of the cloning vectors. Yes, anyone? Ori should be present, okay. Selectable marker, cloning site, cloning vectors, okay. So only two students responding in the group. Restriction site, okay. Okay. So cloning site is a okay, cloning vectors. Okay. Selectable marker for identifying the transformants. Yes, absolutely right. Any other? Kirtini not responding. Ragul not responding. Gopinath not responding. Why? Is this class for only 2 or 3 students, selective student? No, this class for every student, right? Then why only 2 or 3 students responding? I want response from all of you. Yes, TI plasmid in plants and retrovirus in. So this is the okay, fine. Come to the types of cloning vectors. And the first one, as I discussed, that plasmids are the most commonly used cloning vectors, and they are, what are the characteristic features of these plasmids? Yes. So here you can see the most commonly used cloning vectors, that is called plasmids, and these plasmids are circular double stranded. Means what are structures like this? Like this. This kind of plasmids are there. This is the real plasmids. This is what shown over here, a complete circle. No, it is not always complete circle. It is what? It is circular double stranded DNA molecule that occurs as an extra chromosomal materials in the bacterium. That is what the plasmids. Okay, and the most common one is a PBR322. Here, this is the most common one, right? Now here, there was the first artificial cloning vectors constructed in the year of 1977, used in the gene cloning experiment, and that artificial cloning vector is called PBR322. Now here you can see this was constructed with, this is artificially cloning vector constructed in 1977 by Bolivar and Rodriguez. It was constructed by Bolivar. Bolivar is the name of the scientist who constructed this. So Bolivars and Rodriguez who constructed this vector, and it is widely used in the gene cloning experiment. So any cloning vector have to follow some nomenclature. It is not so easy to give the name of any vectors. There are, as like the plant having, for plant naming, there we should have to follow some nomenclature. Similarly, for vector also we have to follow some nomenclature process. So in PBR 322 plasmids, P is always stand for plasmids. The small p, you can see here what I have written, that a small p, this small p is represent the plasmids. Here, now there is a BR. So what is mean of this BR? So BR is nothing but it is the stand for Bolivar and Rodriguez. So BR stand for Bolivars and the Rodriguez. Let me write here the name of that scientist. It's a B for Bolivar, and R for Rodriguez. Okay, so Bolivar and Rodriguez who constructed this PBR. So that BR stand for Bolivar and Rodriguez. Now why it is 322? Now what is this number represent? 322. So this number 322 is the number given to differentiate this plasmid from the other developed in the same laboratory. Like in that same laboratory where this PBR 322 discovered, in the same laboratory there is another plasmid which is constructed like PBR 325. Then there are certain more like PBR 27, PBR 327, then PBR 328, PBR 328. Then there is a PBR 345, PBR 345. So these are the different name of the constructed plasmid which is constructed in the same laboratory, and that's why this number, last number, represent the typical like when discovered first and when discovered later. So later is given the number according to the discovery. Okay, so always keep in mind that number is given to differentiate this plasmid into the other plasmids which is developed in the same laboratory. Clear? So these are the name itself, PBR 322. Is this clear about the nomenclature? Why it is PBR 322? I want response from all of you. Okay. Okay. Now here you can see the complete structure of PBR 322. So as I told you, it was the first artificial cloning vectors constructed in 1977 by Bolivar and Rodriguez. It is widely used in the gene cloning experiment. So any kind of gene cloning experiment, this PBR 322 is generally used. Now here you can also find the different site. Like here you can see this is stand for tetracycline. Then here you can see this is ampicillin, antibiotics. Here's another one. Okay, so this tetracycline contain BAM, H1 and SAL1, and this ampicillin contain PST1 and PVU1. Similarly, there is a EcoR1, CLAV1, hint 2 is there, origin of replication over here, origin of replication over here. So these are the different location in one cloning vector. Okay, now come to the bacteriophage. Now many of you know very well what is bacteriophage. Those virus who infect the bacteria called bacteriophage. So bacteriophages are virus that attack bacteria. So they follow two important cycle that we all know, that is called the lytic cycle and lysogenic cycle. These two cycles followed by the bacteriophage, right? Now these bacteriophages, what are the other characteristic features when we say? So bacteriophages are the virus that generally attack on bacterial cell by injecting their DNA into these cells. Now where the DNAs are present, you can see this is the capsid area, that is the head area. Inside this capsid there is a DNA. So this DNA is going is generally injected into the host cell. So this injected DNA is selectively replicated and expressed in the host bacterial cell, resulting in the number of phages which burst out of the cell. That is what called lytic pathway, which follow the lytic pathway. Right? So this ability to transfer the DNA from the phage genome to the specific bacterial host during the process of bacterial infections, that give a scientist the idea that specially designed phage vectors would be useful tools for cloning experiment. So several bacteriophages are used in as a cloning vectors, but most commonly used are lambda phage and M31. Most commonly used. Yes, let me write here. The most commonly one used is lambda, sorry, yes, the first one is that is called lambda phase, another one that is called the M13 phase. Okay, these are the two most commonly used bacteriophage vector. Okay, now let's have detail about these two phases. When I say lambda phase, lambda phase, lambda phase is double stranded, and it is linear DNA, linear DNA, which having the genome size, genome of near about we can say near about 48,502 BP. BP means base pair. Okay, and it is very sticky and sticky, or we can say the cohesive. It is very sticky, and are referred to as a cos site. It has a cos site. Okay, so why it is called cos site? Because cohesive end site, no? So it's referred as a cos site. Cohesive means sticky. So cohesive end site. Now these sites are important for packaging the DNA into the phase head. That's why this cos site is very important. Now the lambda genome remain linear in the phase head, but within E. coli cell, two cohesive end join to form a circular molecular necessary for replication. So this vector allow the cloning of DNA fragment up to 23 kb. This lambda phase allow the cloning up to 23 kb. This is the size that you have to remember, 23 kb. Kb for kilo base, right? Kilo base. As you know that unit, it is a kind of unit. Kilo base is a kind of unit which is especially designed the length of nucleic acid sequence. So 1 kb stand for 1000 nucleotides. Okay, 1 kb stand for 1000 nucleoside. Sorry, 1000 nucleotide base. So imagine it can create up to 23 kb. Is this clear about the lambda phase? Okay. Similarly, there is a second one, M13 phase. Now, when I say M31 phase is also another kind of vector, it is filamentous phase which infect E. coli having F. pili. That is very important. Okay, this M13 phase is a filamentous phase. Filamentous phase which infect E. coli effects. Like having F pili. Now F is what? That is a fertility site which is called F pili. Oh, it's a pili, not pili. F pili. Okay. Now here its genome is single stranded. This is a single stranded circular DNA. The first one is double stranded circular DNA, linear DNA, but here it is a single stranded. It is single stranded circular DNA, of, and the genome size is 6407 BP, not KB, it's a BP, that is a base pair. Clear? Now here again the very interesting things in this M13 fast bacterium, that is after the M13 fast DNA enters the bacterial cell, it is converted into a double stranded molecule. Right? Initially it is single stranded, but when it inserted into the bacterial cell, it converted himself as a double stranded molecule, that is known as a replicative form, or it is also referred as a RF, replicative form. Clear? And so this replicative form, which replicate until there are 100 copies in the cell, and single stranded copies of genomes are produced and extruded from the cell as M13 particles. That means it create 100 copies, right? So the major advantage of these developing vectors based on M13 are that is its genome size is less than 10 kb in length. So here genome size is less than 10 kb, because in the above one genome size is up to 23 kb, but here is less than 10 kb can also create. Now here in this M13 fast vectors, the RF can be purified and manipulating exactly like a plasmid. So in addition, this gene cloned in M13 based vector can be obtained in the form of single stranded DNA. So the first one, that is lambda fast, is a double stranded linear, but here it is single stranded and circular DNA. So these both are actually the part of, or we can say the type of bacteriophage vectors. Clear? Is this clear to all of you? Though because these two are not mentioned in this, that's why I wrote here on the screen, so it will be very easy for you to recall. Yes? Anyone have a doubt in this bacteriophage? Okay. Another type of vector, the third type, I mean to say, is the third type, plasmids. Right? So plasmid is a combination of plasmid and 200 base pair of lambda fast containing the cos site of lambda fast. So that's why plasmid. Cos means it is a part of lambda, and mids, so it comes from the plasmids. So actually it is a combination of plasmid and the lambda fast, or we can say plasmid and the bacteriophage lambda. Right? So let's have this. So as I told, cosmid, so cos plus plasmids become a cosmid vector. So the term cosmid is a combination of two words. What is combination? cos and mid become cosmid. Now here cos is taken by us and mid is taken from the plasmid DNA. So it become a cosmid. And this was developed by the first time by the Collins and Holmes. This was developed by the Collins. And the another scientist is Holmes. Okay, who developed this cosmids, because it is again the artificial chromosomes. Now here cosmids can be used to clone the DNA fragment up to 45 kb, up to 40 to 45 kb of DNA inserted in it. That means a quite bigger as compared to the lambda fast and the M13 fast. This is quite bigger. Can accommodate the 5 kb of DNA insert in that. You can imagine, see 1 kb, I told you 1 kb means how many DNA, how many nucleotide in 1 kb, how many nucleotide? 1000, right? So it can accommodate up to 40 to 45 kb of DNA, quite bigger in size, right? Similarly, there is another one, the fourth one, that is called yeast artificial chromosomes vectors, or YAC. Now again, YAC it is a linear. These are the kind of vectors used to clone the DNA fragment of more than 1 MB. 1 MB means you understood how much. 1 MB. What is stand for MB? Yes. Yes or no? Tell me. 1 MB stand for what? Mega base pair, or it is a kind of memory unit, megabyte, gigabyte, GB, KB, kilobyte, mega base pair. Very good. So it's a 1 MB stand for mega base pair, and so mega base pair means how many base pair in size. If I say 1 MB means how many base pair, because we know the one base pair, nucleotide base pair. So how many base pair is it? In 1 kb there is 10 to the power 6 base pair, right? So that is interesting, quite interesting, that you have to remember. And because of such huge size, they have been exploited extensively in mapping the large genomes. Like we can say, we know very well about the Human Genome Project. So yeast artificial chromosome vectors are generally used in the mapping of large genomes such as Human Genome Project. And these vectors contain the telomeric sequence and the centromere, and automatically replicating the sequence from the yeast chromosomes. So they also contain the restriction site, enzyme site, and gene that act as selectable markers in the yeast. And that's why this YAC vector is preferred more on the larger mapping of genome project, we can say. Clear? So that is yeast artificial chromosomes. Is this clear? So you have to remember that linear vectors that behave as a yeast chromosome, it consists of sequence for applications, 1 or 2 selectable marker is there. Then there is a telomeric sequence at both the end. Why it is telomeric sequence? Because it is a linear. So linear means it is always like a straight line. So telomeric sequence at both end. Here is the one end, another one end is over here. So at both the end there is a telomeric sequence. So here is tel, here is also a tel, which is called telomeric. Like here you can see when you cut with the balm, then when you straight it, it gives you the straight linear vectors, isn't it? So telomeric sequence is present at both the end. Is this clear about the yeast artificial chromosome? Okay. Now the another one, the fifth type, that is called the bacterial artificial chromosome vector. Now here bacterial artificial chromosome vector, these are a vector based on natural extra chromosomal plasmid of E. coli DNA. This is naturally available in E. coli bacterium, which is called extra chromosomal plasmid. Right? It is naturally available in all the bacteria. So this vector can accommodate up to 300 to 350 kb, 300 to 350 kb, that is called kilobase. So kbs of foreign DNA and are also being used in the genome sequencing project. Means artificial YAC and BAC both are used as a large genome sequencing project. Both can be used for large because they have such a huge their capacity, genomic capacity, and that's why both can be used as a human genome project. So that is YAC and BAC. Clear? You can see the size 300 to 350 kb. Clear? So it have the sequence of replication of origin, as you can see replication of origin over there. Then it control the stability of to maintain one or two copies of vector per cell, quite good in number. Okay, the last one is there, the sixth one, subtle vector is also there. This is the last one. So when I say subtle vector, it is quite interesting as compared to the others also. So subtle vector basically it exists in both cell. It is present in eukaryotic cell and also in E. coli cell. So those vectors who can exist in both the eukaryotic cell as well as E. coli, such vectors are known as a subtle vector. So these these vectors maintain two type of origin of replications. One that is selectable marker gene and one type that function its eukaryotic cell and the another it functions as E. coli cell. Because it is a combination of these two, right? So that's why it have a two selectable marker gene, right? The example for this subtle is like yeast episomal plasmids. The most common example is YEP. YEP. It is written like this only, capital Y, capital E, and small p. So YEP stand for its episomal. Let me write here. Yeast episomal, and P stand for it's a plasmid. Plasmids. Clear? YEP is episomal plasmids. Now here, in case of plant, because this subtle vector can be possible to present in animal also, in plant also. So in case of plant, naturally occurring plasmid of bacteria, that is called Agrobacterium tumefaciens, called TI plasmid, has been suitably modified to function as a vector. So most of eukaryotic vectors are subtle vectors. So always keep in mind that most of eukaryotes, this is the statement which you have to remember, most of eukaryote eukaryotic cell or eukaryotic vectors are shuttle vectors. Shuttle vectors. This is a statement which you have to remember. Yes. Is this clear? Okay. Another vector is also there, like animal vector. As we studied the plants, animal vector is also there. So when I say animal vector, so animal and plant vector, so some vector based on the simian virus SV40. Okay. So DNA fragments from the animals are cloned in E. coli and then integrated with the different viruses to be introduced into the host animal cell. For example, like SV40. Now this SV40 is called the simian virus. Simian virus 40. Okay, and was used in the first cloning experiment involving the mammalian cell. Mammalian, all the mammalian cell, simian virus generally use such vectors to transfer. So it's the first mammalian cloning vectors that is called the simian virus. Yes. Another one, plant vector. So in plant vector, that is a Ti plasmid. So two plasmid, namely Ti plasmid and Ri, are present in Agrobacterium tumefaciens and Agrobacterium rhizogenes. So you have to remember that Ti means it's Agrobacterium tumefaciens. Tumefaciens. And the another one is Ri plasmid. Ri stand for it's a Agrobacterium rhizogene. Small r consider. So rhizogene. These are the two plant vectors that are considered the natural genetic engineer of the plants. Okay, so this is all about the vectors, characteristics of what is vector, characteristic vectors, and the different type of vectors. Yes, anyone have a doubt in this? Like what is vector, what are the characteristics of vectors, what are the types of vectors, and what their size and all everything. Anyone have a doubt in that? Okay, now I'm going to give you break for 5 minutes. Yes, want a break or no break? Okay, and said break. Okay, Sajana is also responded break. Okay, so yes, you all can take a break for 5 minutes. We will start our session again after 5 minutes. Okay, so please take a break. Hello. Hello. Okay. So now the break is over. Please on a front camera. Yes, Sajana and Kirtini. Okay. Okay, Sajana back. Okay. And okay, fine. So Ragul and Gopinath, please respond it. Ragul and Gopinath. Okay. Now we complete the cloning vector. Another one, that is competent host. Now once you select the cloning vector, you require competent host for transfer of these cloning vectors. That is vector mediated gene transfer. So what is this? Vector mediated gene transfer. So first of all, competent host is essential for transformation with recombinant DNA. Now when I say transformation is what? It is a process by which a cell take up naked DNA fragment from the environment and that incorporate into its own chromosomal DNA and finally express the trait controlled by the incoming DNA. Right? So this is what called the transformation with a recombinant DNA, or we can say the competent host, and DNA mediated gene transfer. Right? So many kind of host cell are available, like we can say the E. coli cell, then there is a yeast, animal, plant cells are available for genetic engineering, and the kind of host cell to be used mainly depends on the aim of cloning vectors experiment, like what kind of aim of particular experiment. For the expression of some eukaryotic proteins, or we can say eukaryotic cell, may be preferred host, or it is basically have been used extensively for functional expression of eukaryotic gene because they offer several advantages. What kind of advantage? You know very well that yeast are the simplest eukaryotic organisms, like bacteria, and like bacteria are single celled and genetically well organized and manipulate because bacteria or yeast generally grow in any suitable media, right? So it's very easy to grow and multiply, or we can say the manipulate, right? So they can be grow readily in both culture vessels, or we can say large scale bioreactors. Bacteria and yeast are very prominent. It can grow in a culture laboratory like in the petri dish you can grow, or in a very large bioreactor you can grow. So plant and animal may also be used as a host in the gene manipulations experiment and for protein expression either in the tissue culture or any cell in the host whole organism to create a genetically modified plant or animal. Since we are talking about the DNA, so since the DNA is a hydrophilic molecule, it can not pass through the membrane, so the bacterial cell must be made available to take up the DNA. And this is done by treating with the specific concentration of divalent cation, I can say calcium. Okay, So when you treat that particular DNA with a specific concentration of divalent cation, such as calcium, which increase the efficiency with the DNA enter the bacterial through the pores in its cell wall, right? So this recombinant DNA or RDNA can then be forced into the such cell by incubating the cell with the recombinant DNA on ice, and followed by placing them in a 45 degrees Celsius heat soak and then putting them back on the ice. So this enables the bacteria to take up the recombinant DNA. As you can see, it is shown in this figure that how it can uptake. So that means of there is a particular process required, right? So these vectors, that is what the DNA mediated gene transfer, that is what called. Clear? So what is important for first of all, the capable DNA that need to treat them with a specific concentration of divalent cation. This is very important. The extracted DNA are treating them with a specific concentration of divalent cation like calcium. Why we need to treat them with the calcium? Because calcium increase the efficiency with the DNA, so that the DNA enter the bacterial cell through the pores in its cell wall. Very easy. When you concentrate them, treat them with the concentrated calcium, it will be very easy to pass that DNA into the bacterium cell wall. Now here the recombinant DNA can then be placed them in 45 degrees Celsius. So 45 degree, you know that 45 is quite in relevant for bacteria, but when you placing them bacteria at 45 degrees 42 degrees Celsius, it is called the heat soak, and then putting them back to the ice. So first is a hot treatment, and then followed by there is a cold treatment. So this enables the bacteria to take up the recombinant DNA inside it. This is what the transfer of such DNA into the bacterial cell is called the transfection. This type of method is called transfection. Clear? Transfection, is this clear to everyone? Yes. Initially, a very hot soak like 45 degrees Celsius, and then followed by cold treatment. Clear? So this process is transfection. Another method of competent host is the direct or vectorless gene transfer. This is the one-way method, that is a DNA transfer. DNA, here what we did a DNA mediated gene transfer. The another method is called the direct or vectorless gene transfer, right? Direct and vectorless gene transfer. So it is a process of gene transfer into the host cell without using a vector. So there are some artificial techniques, okay, through that you can directly inject that desired gene into the host, and this is possible by some important method. And one of the great method is over here, like micro injection. You can see here the micro injection. This is a direct, or we can say vectorless gene transfer method. Clear? Now what is in this micro injection? So as the name itself indicates, micro injection means injection which is very small in size, but their work is to transfer the desired gene. So in this method, foreign DNA is directly injected into the nucleus of animal cell or plant cell by using a micro needles or micro pipettes, right? So it is basically used in oocytes, egg and embryo transfer. And this technique, micro injection, Alex Jeffrey is the name of scientist So Alex Jeffrey of Human Genome Center, Michigan University of USA has cured mice that inherited a neuromuscular disease, which is like a muscular dystrophy of humans. Okay, so he used these techniques of micro injections for direct transfer of desired gene into the nucleus. So the one is micro injection. Another one is electroporation. Electroporation. Now what is this electroporation? It is also electroporation is a formation of temporary pores in the plasma membrane of the host cell by using a lysozyme and calcium chloride treatment. Lysozyme and calcium chloride treatment. Clear? So here you can see this is how the electroporations, like suppose this is a plasmid DNA. Okay, so electrocompetent cells on ice and then transfer to cuvette into the ice and then give an electric shock to this plasmid DNA. Then transfer to the host cell, transformed host cell. So you can see it's similar to. So it is what actually it provides some electrical shock to create the pore, right? So through that pore, that part of interested gene can easily go because there is a minute pore, so it can easily pass into the nucleus. So that is one another method of direct gene transfer that is called electroporation. So that electroporation is possible only in the presence of certain enzymes. That enzyme is called lysozymes and calcium chloride, and there is a calcium chloride. Okay, so this is a second method of competent host direct gene transfer. Now the third one is a chemical mediated gene transfer. Chemical mediated gene transfer. So this kind of method, certain chemicals are used, and that chemical name is polyethylene glycol. PEG, polyethylene glycol. Okay, so polyethylene glycol, basically this method used to transfer the DNA into the host cell. How? So you can see the technique that is called the chemical mediated gene transfer. Just a minute. So here you can see in this chemical method, polyethylene glycol, that is what called PEG, polyethylene glycol, and polyvinyl alcohol is used. Polyvinyl alcohol. Okay, these two chemicals are used because it is a chemical method, right? Now what is the process of this chemical mediated gene transfer? So the plant protoplast is placed in the medium rich in magnesium. To this, the plasmid DNA with the desired gene is added. So when you keep that plant cell into media rich having magnesium, ample amount of magnesium is there, so that what happens, the plasmid DNA with the desired gene is added. So after this, polyethylene glycol is added and the pH is maintained at 8. So once the pH is maintained, the protoplast is given a 5 minutes heat treatment and then placed in the ice before the addition of DNA. This increases the frequency of the transformation. And after some period of incubation, the polyethylene glycol is reduced and the calcium enzyme increase. This helps the frequency increase the transformation frequency. This is what the chemical method. Is this clear? Hmm? So here you can see this is a polymer or polymeric nanoparticles are there. This is a DNA which is your design. So when you mix with this DNA with a polymer or polymeric nanoparticles, so it become a positive charge over there, and then it entered into the cell mediated gene transfer. So you can see how it perform the entire process. Another one you can see the biolistic method, or we can say the gene gun method. Biolistic method, or we can say the gene gun method. Now here, basically for this kind of work, 1 or 2 tungsten or gold particles coated with the DNA is used for transformation. So this entire gun, see here in this picture, this is a gun. So this gun basically called the gene gun method or biolistic method. So basically this is basically coated with the tungsten or gold particles. Okay. So these particles are accelerated at high velocity, and thus this gene enable it to enter into the host cell. Like here you can see, like here this in this third picture, see how the gene is introduced into the host cell. Right, so this is the biolistic method. Okay, another method is lipofaction. Okay, this is also another method of direct gene transfer. So lipofaction, basically the desired DNA is introduced into the cell with the help of liposomes. Liposomes is a kind of we can say vectors. It is not actually vector, but it is a kind of organelles through which it is going to be transferred. So this method can be used for animals as well as the plant of lipofaction. Okay, so the entire interest of gene is introduced into the host cell with the help of liposomes, and this method can be used for animal as well as plant. So you can see here, this is a transfection reagent, and this is a plasmid DNA that you want to introduce into the cell. So there is this in transfections, I mean to say the liposomes incorporate that DNA, and that DNA can easily enter into the cell. Now another one is a macro injection. Now micro injection we know, now this is a macro injection. Now what is this macro injection? So basically macro injection, DNA plasmids are injected into the lumen of the fluorescence. This is interesting. That is lumen of the inflorescence, and using a hypodermic syringe. Yes, by using a hypodermic syringe. That is a macro injection. Okay, so these are just a method that you have to remember, that how this gene is directly transferred. So method and technique you have to remember. Is this clear? Okay. So what we studied today? Today we studied about the most common principle of this biotechnology chapter, biotechnology, that is based on four important facts. One is, that is important extraction of DNA, first of all, then requirement of vectors, then requirement of competent host. Clear? So we studied today, that is cloning vectors, what are cloning vectors, what are the characteristics of cloning vector, what are the types of cloning vectors. Then we studied about the competent host, which includes how actually, what are the methods that we can use to transfer the desired vectors or the gene directly into the cell. So If you are using the DNA mediated gene transfer, then it need a certain special vectors. And if another method is direct gene transfer method, so you can use the different tools as well as enzymes or as well as the organelles to transfer the desired gene into the host, and that's why it's a competent host, right? Now I'm going to ask you, if you have any doubt, then please tell me again. Otherwise, we will move on assisted practice kind of questions. We can try a few questions based on the today's topic. Yes. Tell me fast. Any doubt? Okay. Okay. So Anne responded. I want a response from all of you. Kirtini, Sarjana. Okay. Gopinath. Okay. If no doubt, then please read this question. This is the questions. Given below figure refer to the recombinant DNA technology, that is rDNA. Identify A, B, C and D. So here you can see this is the below, means this is the figure over here. To identify what is A, what is B, what is C and what is D, you have to identify. Clear? Now I'm going to give you the option. Some option over here. Now here what you have to identify, you have to identify the enzymes used to cut the foreign DNA, to cut the vector plasmids, to insert the DNA. All this A, B, C and D is given to you. Okay, so you have to identify which one is the enzymes. Now here option is given. A stand for restriction endonuclease, restrictions endonuclease, DNA ligase and transformation. Exonuclease, endonuclease, hydrolase, transductions. Another one is given as, so four option is given to you to identify the enzymes and their roles based on the diagrams which is given. Yes, tell me the answer for this question and responded. I want response from all of you. Please responded. Hope I'm audible to all of you. Yes or no? If I'm audible, then please respond it for this answer. Kirtini, please respond it. What are you doing? Kirtini, why are you not responding? Kirtini, am I audible to you? Okay. So responded, please. Gopinath. Okay. So the right answer for this question is A. A is the right answer. That is, you can see here in this picture. Here you required restrictions endonuclease. Okay. These restrictions endonuclease is a kind of enzymes who going to cut both foreign DNA as well as the plasmid vector at a specific point, right? So therefore you need a restrictions endonuclease because endonuclease is going to cut at a specific point. Not required exonuclease. Why exonuclease means it is going to cut at the end towards the periphery, right? Towards the both the ends. So here it is not required. That's why A and B are both are same. That is restriction endonuclease and restriction endonuclease. Then here, now this is your foreign DNA. When you introduce into the vector, there is a requirement of what? What you required? You required ligase, a kind of enzyme to join this, right? So here is a requirement of ligase. So as you can see the answer, the next is DNA ligase. That is also called the DNA ligase. And when thereafter you need to transfer this entire vector into the competent host. So what is that E? E is what? E is called a kind of competent host, and that method is called the transformation, because when you transfer that vector into the host, it is called a transformation, right? Yes or no? So that's why A is the right answer. Now the next question is polyethylene glycol method is used for biodiesel production, seedless fruit production, energy production from the sewage, gene transfer without a vector. Tell me, polyethylene glycol method is used for what? Kirtini responded. Now this is very simple question. Yes. Okay, just a minute. Okay. So only Anne responded. I don't know what others doing. Kirtini, Gopinath, Ragul. Why not responding? I'm asking something, right? You have to just respond on these questions. Is that questions you feel difficult? Kirtini, Kirtini, what are you doing? Kirtini, please respond it. Why are you not responding? Okay, finally responded. Yes. So the right answer for this question is very simple. It's a gene transfer without a vector, because vector mediated gene transfer is DNA mediated, right? Something. Okay. Another one. PBR plasmid is named after what? After its discovery, its genetic material, its mode of replication, or the source from which it is obtained. Okay, responded. Kirtini, Kirtini, please responded. Okay, so it is named after the discoverer. So that is what PBR plasmids. Okay, the another question. The next question, please. Okay. In the plasmid PBR322, the term B and R reports which of the following? It is name of the scientist, name of the restrictions into nucleus enzymes, name of the cities in the USA, name of the strain of bacteria. Excuse me. So it is the name of scientist. Very easy. Okay. So this is the summary what we studied, that is properties of good vectors. So good vector replicate autonomously. Excuse. Should be able to carry the genome into the host cell, less than 10 kb in size, high copy number, easy to translate and purify, should be able to integrate with the DNA insert, easily introduced into the host, most suitable markers, and unique target site for restrictions enzyme. These are the classical properties of good vector, and this is what the different types of vector what we studied. So there are FAS like lambda and M13, cosmids, then transposons, then plasmids, then YAC, BAC, subtle vectors, and plasmid, which include the PBR 322 and TI plasmids. So this we have already studied in details, right? Now we completed this concept, two concepts today. One is cloning vector and its properties. Another one is a competent host.